Hello, I present you RNA Tailor, a new tool designed to precisely identify the alternatively spliced transcript diversity of a target gene from long-read RNA-seq data, such as PacBio or nanopore data. It analyzes the data without annotation, and it has the advantage of not being biased by previous knowledge and to be usable on non-model species. In order to run RNA Tailor, the user needs to provide in input the ensemble ID of the target gene and the long-read RNA-seq dataset in which the gene is expressed. So the first step of the analysis is to perform a read selection in the dataset to extract read of interest. It is performed either with Minimap2 or MegaBlast. Once the read is selected, the next step is to align them to the reference gene sequence with the spliced aligner exonerate. So that's the main advantage of RNA Tailor compared to similar tools, because exonerate provides a precise alignment thanks to its semi-global spliced alignment method. So the alignment is done in two passes. The first pass aims at spliced align the reads to perform pruning of false positive reads identified based on their alignment. After this step, the remaining reads are auto-corrected using the tool ISMB. The aim is to drastically improve the quality and accuracy of the mapping. Then the reads are realigned with the splice aligner to the reference sequence of the gene. So at this point of the analysis, we can consider that the reads that are in the batch should share a common splice structure as they originate from the same gene. Therefore, we apply a filter based on splice junction inconsistency found in the reads, as an inconsistent splice structure for a read would mean that it is a contamination of our batch. So it is done in two passes as well. First, we identify inconsistency, and after this identification, we perform our misalignment correction method, as inconsistency in the read can result from error in the alignment process. So to try a resolution of this inconsistency, we perform local alignment with exonerate of a subpart of the read. After this realignment correction, if the splice structure of the read is still considered inconsistent, then it is discarded from the analysis. So now we work on reads with a consistent splice structure, but we still have an issue with the precision in the positioning of the exonic junction on the genomic reference. That can still be affected by sequencing error and alignment bias. So to account for this, we apply a majority vote method to select the most supported position of each spike junction on the genomic reference. So as a result of border smoothing, here, we have a batch of reads with a homogeneous structure and precisely defined genomic position of splice junction. We can now process to transcript clustering, and we do that based on intronic structure of the predicted transcript. So it has been chosen to account for long-read tendency for incompleteness that makes difficult the exact characterization of a precise nucleotide start and stop. So each predicted isoform is kept only if it is supported by at least two reads, and if not, it is considered as a false positive. The results of the isoform prediction is exported in standard format, such as GFF, TSV, and NixLS6 spreadsheet, giving a clear and simple overview of the alignment structure. So here is an example of this spreadsheet for gene of the mouse. So here on the left, we can see each line corresponds to a read name, And here each colored column corresponds to a segment of the read aligned on the reference sequence of the gene. And if a GFF file is provided, then known exon can be highlighted. So here, for example, we see that if a column is full, like here, it might show a constitutive splicing dynamic, or if it is not full, it might show a possible cassette exon. So we tested the prediction result of RNA Taylor with against two other tools, Freddy and Flair. And to do so, we tested the predictions of each tool for 19 mus musculus genes with the splicing dynamics. So the reads were originated from the public dataset of Heiner Aster, and we tested the predictions against the annotation data of each gene, and we calculated how many known reference isoforms were found by each tool. So as a result, we can see that RNA Taylor found the 25 known isoforms against 22 for Flair and 7 for Freddy. So RNA Taylor is here achieving either similar or better results without annotation, and that suggests very promising results, mostly on non-model species. So this work was supported by Heiner Aster and the JDR beam, and the link of the tool that is available on the GitLab is on the poster. Thank you very much.